Welcome everyone, and thank you for attending Stemina's Exhibitor Hosted Session at the 2021 Annual Meeting for the Society of Toxicology. My name is Jessica Palmer, and I am the Associate Director of Toxicology at Stemina Biomarker Discovery. Today I'm going to talk to you about application of our Biomarker-Based In Vitro Assays, devTOX quickPredict and Cardiotoxicity for Toxicity Screening. There's a lot that I'd like to cover in today's talk. First, I'm going to discuss briefly Stemina's approach for developing biomarker-based assays, and then I'm going to do an overview and a few application examples for each of our assays. For those of you who do not know about Stemina, we were founded in 2007 and our laboratory is in Madison, Wisconsin. We focus on mass spectrometry and stem cell culture, and we really look to develop human stem cell models of toxicity and disease. We also have a sister company called NeuroPoint-DX, which is focused on developing diagnostics for neurodevelopmental disorders. Specifically, right now we're working on and offer a diagnostic for autism spectrum disorders. So the approach that we take at Stemina is we really felt that human pluripotent stem cells offered a unique opportunity to evaluate chemical toxicity using a human endpoint, with the goal of really eliminating species-specific differences in toxic response. One of the benefits of using pluripotent stem cells is that you can really differentiate them into all cell types from the body that can function like their in vivo counterparts. And the cells, or cells derived from pluripotent stem cells, are really applicable to high throughput systems. Our devTOX quickPredict assay focuses on using embryonic or induced pluripotent stem cells in their undifferentiated state, whereas our Cardiotoxicity assay is an assay for adult cardiotoxicity and uses cardiomyocytes that have been derived from induced pluripotent stem cells. So we combine this human pluripotent stem cell biology with LC-MS-based metabolomics to develop assays that would allow our clients to understand compound risk profiles faster. We're able to provide results in as little as 2 weeks. We look at multiple exposure levels using a minimal amount of compound, and hopefully all at a cost savings compared to the standard in vivo assays that you would have to do. So the two assays that we're going to discuss today, as I mentioned, are our devTOX quickPredict and our Cardiotoxicity, and both of them really provide an exposure-based prediction of toxicity potential using a targeted metabolomics in human pluripotent stem cells and differentiated cells. So devTOX quickPredict has been optimized for use with either human embryonic or induced pluripotent stem cells. The embryonic stem cell line that we use is one of the original ones derived by James Thompson back in 1998. And then we use a commercially available iPS cell line that has been reprogrammed to that embryonic-like state. And both of these cells have the ability to self-renew and are pluripotent in culture. What we're measuring is not the intracellular metabolome, but really what we call the secretome. So what we measure is how the cells are interacting with their media. And so when we were developing the assay, we used a training set of 23 well-characterized compounds that had known human developmental toxicity outcomes. And we used them to identify the most predictive metabolites so we could go from an untargeted metabolomics approach to a targeted approach. And through these studies, we identified ornithine and cystine as being highly predictive of developmental toxicity potential. Both of these metabolites are essential for normal embryonic development. Ornithine is secreted by the cells into the media, and it can decrease or increase with toxicity, although decreases are what are most commonly seen. And biochemically, it is involved in the urea cycle, polyamine synthesis, and pyrimidine synthesis. In contrast, cystine is actually a media component, and it is consumed by the cells from the media. So when they're happy and healthy, the cells will use at least 75% of that cystine that's in the media. However, when you treat them with a developmental toxicant, what you'll see is an increase compared to the controls, indicating that the cells stop utilizing cystine from their media. And its main function is in the cystine-cystine redox cycle, which is a modulator of oxidative stress in the cell. Now, both of these are essential for normal proliferation and differentiation during embryonic and fetal development, and you can imagine that if either one of these processes is disrupted, it can lead to malformations or embryonic death. So the process that we use is we'll culture the cells in 96 well plates and look at 8 exposure levels of every test article. Each plate will have a positive and negative control to ensure that the assay is performing within the specifications of how we designed it. And we'll collect the spent media and process it for LC-MS analysis, where we measure specifically ornithine and cystine, as well as internal standards for these two metabolites. This allows us to use the cells for additional downstream processing, and our standard endpoint is cell viability, but we can also collect cells for additional analyses if there's a need. And what we'll do is we'll take the data from these two metabolites and from cell viability and fit them with dose response curves to really understand at what point is a chemical shifting from non-developmentally toxic to developmentally toxic. So if you look at that a little closer, the assay is really designed around the idea that toxicity is a function of exposure. And so we use an 8-point concentration curve to look at what point does a compound alter metabolism that's indicative of developmental toxicity. And so what we want to know is when they cross this a drug or chemical cross this developmental toxicity threshold. This threshold was set using that original 23 compound training set and looking at the response of the compound in the assay compared to where it is known to be developmentally toxic in vivo. And using this dose response curve from the OC ratio, we can identify what we call the developmental toxicity potential concentration, which is where that OC ratio crosses that threshold. And then this data could be compared to your cell viability dose response curve to understand the mechanisms of the chemical in the assay and whether or not it is causing changes in the cells independent of cell viability or concurrent with cell viability. And using this DTP, we're able to really identify non-developmentally toxic exposure windows as well as developmentally toxic exposure windows. And we've done a number of studies internally and externally to look at how accurate do these two cell types predict developmental toxicity. And the chemical set that we've used is really quite diverse and includes pharmaceuticals, agrochemicals, cosmetics, industrial chemicals, environmental chemicals, and we compare this data to the published human results when possible, but most often it's going to be your in vivo rodent or rabbit results. Now, to assess the performance of the assay, we've developed 2 different prediction models. The first is when if there is in vivo data available, we'll score this DTP concentration against the Cmax. And so a compound that has a DTP that's greater than 10 times the Cmax would be predicted to be non-developmentally toxic, whereas a compound with a DTP concentration that's less than 10 times the Cmax would be predicted to be developmentally toxic. Now, much of this data or exposure data isn't always available during early compound development, and it's not always available for things like agrochemicals and industrial chemicals. And so in those cases, we use the training set to set a threshold to determine at what concentration can we separate developmental toxicants from non-developmental toxicants. And that threshold was optimized to be 65 micromolar. So if you look at all the chemicals that we've tested in both embryonic stem cells and induced pluripotent stem cells, you can see that the assay performs quite well and has greater than 80% accuracy, with sensitivity up to 88 and 90%, depending on the cell type. The specificity was slightly lower in human embryonic stem cells than it was in the induced pluripotent stem cells. And this isn't necessarily because the cells are predicting a drug or chemical differently, but because there's a difference in what chemicals have been tested in which assay. But I think one of the important things to take away is that the assay has a very high positive predictive value. And so when a chemical is predicted to be positive with either cell type, you can be confident that it's going to be positive in vivo. We've looked at the different, I've taken the data set and separated them into different chemical classes to see, to demonstrate that the assay can be broadly applicable across these different chemotypes. And you can see that, the balanced accuracy is from 80 to 94%, depending on the use, with sensitivities of greater than 85% with these data sets. And I have a poster at SOT this year that looks at each of these classes a little bit deeper and looking more closely at mechanisms of action and whether the assay is able to accurately predict specific types of pharmaceuticals. And if you're interested on the agrochemical side of things, one of our sponsors at Syngenta is also doing a poster on the use of the assay with agrochemicals. So devTOX quickPredict is currently being used for decision making by multiple industries. And I'm just going to go through a few examples of how the assay is being used. In particular, I'm going to cover how the assay was applied using the EPA's ToxCast program with our publication that came out with the EPA about a year ago now. I'm also going to touch on one of the examples of use that we developed specifically for gaseous compounds in the chemical industry, as well as some work that we've done with one of the tobacco companies. And that research was actually just published this last fall with Imperial Tobacco. But it's also been used in the cosmetics and fragrance industries to look at the different developmental toxicities of chemicals and make decisions on, whether those compounds are going to be developmentally toxic. So there's a lot of data from the EPA ToxCast study that we did, and it really encompasses over 5 years of work looking at 1,066 chemicals or 65 compounds that were either screened at a single concentration or using the concentration response, which is the standard devTOX quickPredict outline. And so what we did is we screened the chemicals and then sent the data back to the EPA, where they processed it using their ToxCast pipeline. And there were a number of different QC steps along the way that are all discussed within this publication from last year. But what we found, and what the major findings were from the study, were that of the 1,065 chemicals, 19% of them were predicted to have developmental toxicity potential. The assay performance, when it was compared with the in vivo animal models of human prenatal developmental toxicity, had an accuracy ranging from 79 to 82% accuracy, with a very high specificity of greater than 84%, depending on the model used. The sensitivity for the EPA study was lower than what we've seen with our internal work, likely due to differences in the chemicals that are tested and the concentration ranges, as well as the broad range of chemotypes that were tested that we may not have included previously. But what we found is that sensitivity really improved as we developed more confidence in the in vivo models that we were using. Now, because of this large data set, they were able to take the data and look at positive and negative associations with other biochemical data sets within ToxCast to really understand what is the biological domain of the assay, where was it predicting developmental toxicity, or what mechanisms was it missing. And what they found was that the assay was sensitive or predicted chemicals to be developmentally toxic for chemicals that modulated receptor tyrosine kinases, as well as associated downstream kinases and phosphatases. And these kinases are known to regulate cell growth, differentiation, and survival. Where it was found to miss developmental toxicants was with calcium second messenger signaling, as well as nuclear receptor mediated gene expression, with the exception of NR3C1. And when we looked a little deeper in the data, we found that many of these receptors were not actually expressed by the cells, so it would be difficult for them to be picking up any of their effects. And these results are consistent with the applicability domain evaluation that I did using pharmacological classes. And this is some of the data that's presented on poster 108. In the next example, I'm going to discuss how devTOX quickPredict was used to screen and compare the developmental toxicity potential of nicotine products. And this research was recently published in Current Research in Toxicology with Imperial Tobacco this last fall. So what we did is we looked at 7 different samples that were prepared from reference cigarettes, a heated tobacco product, a hybrid product, and 3 e-cigarette products. And the samples were generated by bubbling smoke and aerosol up through PBS, which then traps a number of the harmful components of that smoke. Now, these aerosol and smoke bubbled samples could then be applied to the cells, and we looked at the developmental toxicity potential at concentrations up to 10% bubbled PBS. And so we wanted to really compare the effects of tobacco-containing and tobacco-free non-combustible nicotine products to the reference cigarettes to see if there was any difference in their potential to cause developmental toxicity. What this study found was that combustible cigarettes and heated tobacco products caused a change in the OC ratio and iPS cell viability, whereas the hybrid product and e-cigarettes did not impact either of those endpoints. Additionally, the 2 reference cigarettes caused changes in the ratio and viability at lower concentrations than the heated tobacco product, which would be expected. When you look at the nicotine content of the samples that were tested, you can see that all of them were tested at very similar concentrations of nicotine on a micrograms per mil basis, and that the changes we're seeing in iPS cells are really independent of that nicotine concentration, indicating that they're being caused by other trapped constituents in the bubbled PBS. And what this study demonstrates is that the devTOX quickPredict assay could be used for the preclinical assessment of non-combustible nicotine products. In my third example for applying the devTOX quickPredict, I'm going to go over briefly some results from a study that we did where we modified our devTOX quickPredict culture system to be able to treat the cells with gaseous or volatile chemicals. And so this study was conducted in 3 parts, and I'm really going to focus on the results from how the cells responded to those gaseous chemicals. But briefly, in order to culture the cells and expose them to these types of chemicals, we had to develop a system where we placed the culture plate inside a Tedlar bag and then filled it with a specific amount of air 5% CO2 mix, which is similar to what the air content is of the incubator. And then these bags were then placed into the incubator for culture. So first we wanted to see, did culturing them like this change the cell's metabolism or growth, and did it change their response to treatment? And both of those studies showed that we could use this type of culture system and it did not change anything with how the cells were growing or responding to compound. So once we knew that, we wanted to see, okay, do the cells respond to known developmental toxicants when they're treated in the gaseous form? And so we looked at 2 toxicants that had caused developmental toxicity in vivo. Those would be acetaldehyde and hexafluoroacetone. And then we looked at 2 chemicals that had not caused developmental toxicity, HFC 245-FA and 236-FA. And what we found was that the chemicals that produce developmental toxicity in vivo caused developmental toxicity in the assay at much lower concentrations than where any type of response was observed with the non-developmental toxicants. And you can see that these are being expressed as parts per million, so it's the amount of chemical that was put into the bag. And you can see that there is at least 100-fold difference between these 2 classes of chemicals. And so we were able to really show that you could use this assay as a human relevant endpoint for chemical prioritization and risk assessment of gaseous and volatile chemicals. The last example I want to talk about today for applying our devTOX quickPredict assay is how it can be used in conjunction with the new ICH S5 guideline for developmental and reproductive toxicity testing of human pharmaceuticals. This guideline contains a number of scenarios where qualified alternative assays, such as devTOX, could be used in place of or in conjunction with the standard in vivo EFT studies. One of those cases is for drugs that are expected to cause malformations and embryo-fetal lethality based on their class or mechanism of action. In this case, if a chemical or drug is tested in the alternative assay and it gives a positive result, that drug could then be considered positive for malformations and embryo-fetal lethality without any additional in vivo testing. Whereas if it comes back negative, then you would go on to do your definitive EFD studies. To summarize what I've discussed today for devTOX before moving on to our cardio assay, I hope that I've been able to show you that devTOX is an accurate and selective assay for predicting developmental toxicity potential, and that it is applicable to a wide range of samples or chemotypes, anything from your small molecule pharmaceuticals to gaseous and volatile chemicals. Additionally, I wanted to show you how the assay can be customized to meet the needs of our clients. So what's next? We are currently working on incorporating in vitro to in vivo extrapolation so that we'll be able to predict the in vivo dose for a given DTP concentration from the assay. And the EPA is working on a second paper using a machine learning approach to develop predictive models that include the devTOX quickPredict assay and other in vitro data and in silico models from the ToxCast datasets. Moving on to our Cardio quickPredict assay, the work that I'm going to present today was published last year in Toxicological Sciences. The Cardio quickPredict assay was developed using a series of non-targeted and targeted metabolomics experiments using the I-cell cardiomyocyte squared cells from Fujifilm Cellular Dynamics. To develop the assay, we used a diverse set of 81 cardiovascular and non-cardiovascular drugs that had known human cardiotoxicity outcomes. These 81 drugs can be separated into 52 cardiotoxicants, which either cause structural or functional cardiotoxicity, and 21 non-cardiotoxicants. And I'm not going to go through today all of the different studies that we did to identify the metabolites in the assay, but if you're interested, please contact me and I'd be happy to give you more information. Through this series of experiments, though, we did identify 4 metabolites as being predictive of cardiotoxicity. Those 4 metabolites were lactic acid, arachidonic acid, 2-deoxycytidine, and thymidine. The process that we use for the Cardio quickPredict assay is very similar to the devTOX assay. We plate the cardiomyocytes in 96-well plates and expose them to 8 concentrations of a test article. And again, each plate includes positive and negative controls to ensure that the cells are metabolically responding to compound as we would expect them to. We then collect the spent media and use a different targeted LC-MS method from our devTOX assay to measure the 4 metabolites of interest as well as internal standards specific to those metabolites. And then we use the cells to measure cell viability. We'll then combine all of this data and use dose-response curves to predict the concentration where a compound is predicted to be cardiotoxic. The Cardio quickPredict prediction model is a composite of these 4 metabolites and cell viability. Arachidonic acid and lactic acid are both used in a ratio with cell viability, whereas thymidine and 2-deoxycytidine are used by themselves. During development, we set prediction thresholds for each of these metabolites based on the response at 10 times the Cmax for all 81 drugs. And these different thresholds can be balanced for accuracy or sensitivity depending on the stage in which you'd like to employ the Cardio quickPredict assay during drug development. These metabolite-specific thresholds are used to determine a prediction distance, which is the measure of cardiotoxicity potential for each metabolite at each concentration tested. And what this is you're looking at the difference in the response in the cells at a given concentration from that prediction threshold. These metabolite-specific prediction distances are then combined into a composite prediction distance based on the maximum response at each concentration. So for example, the maximum response for this compound at 1 micromolar was obtained in the viability over lactic acid ratio. And so this is the composite prediction distance that's used in the prediction model. So at every concentration, your prediction model or composite prediction is going to be based on the most sensitive ratio. This composite prediction model is then used to determine the cardiotoxicity potential concentration, which is the point where the composite prediction distance dose response curve crosses the cardiotoxicity threshold. So at concentrations prior to this, a compound would be considered non-cardiotoxic, whereas the concentrations after this dose response curve crosses the threshold, we consider that to be our cardiotoxic exposure range. As I mentioned, the prediction thresholds for each metabolite in the composite model can be trained or optimized based on when the assay will be used during drug development. The two examples we provided in our publication were a sensitivity and a balanced accuracy trained model. For the sensitivity trained model, you can see that the sensitivity of each metabolite ranges from 62 to 75%, with a combined sensitivity of 90%. But by training it for sensitivity, you're giving up a little bit in specificity. Whereas when you train the model for balanced accuracy, your sensitivities of each metabolite range from 52 to 65%, with a combined sensitivity of 83%, but your specificity is 90%, with a balanced accuracy of 86%. The metabolites that make up the quickPredict prediction model have key roles in modulating oxidative stress and mitochondrial function and replication, and each metabolite is related to known mechanisms of cardiotoxicity in vivo. There are a broad range of response types observed for each biomarker, which really indicates that the cardiotoxic drugs are affecting different components of the metabolic pathways these biomarkers are involved in. And each metabolite is responding to cardiotoxic exposure independent of changes in cell viability. Lactic acid is a media component, but it's not actually used by the cells under normal conditions. So when an increase in lactic acid is observed, it's because the cells are actually secreting it into the media. This increase is the most common effect that's observed. However, there are a few drugs that actually cause a decrease in lactic acid relative to the controls, which indicates that the cells are actually taking lactic acid from the media internally. This increased secretion reflects a shift from oxidative phosphorylation to anaerobic respiration and glycolysis. And this shift is a hallmark of mitochondrial dysfunction. And this lactic acid readout in the assay provides insight into the state of the cardiomyocyte's mitochondrial energy metabolism. Elevated levels of lactic acid are associated with cardiotoxicity, especially during cardiac ischemia and heart failure. Similar to lactic acid, arachidonic acid is a media component that's not being utilized by the cells under normal conditions. So it's being secreted by the cells into the media. Arachidonic acid can both increase and decrease with toxicity, indicating that there are multiple mechanisms of toxicity being observed here. It is important for lipid transport, lipid metabolism, and fatty acid metabolism. And activation of the arachidonic acid cascade due to redox state unbalance has been shown to contribute to the pathogenesis of cardiovascular disease. In contrast to arachidonic acid and lactic acid, 2-deoxycytidine and thymidine are not present in the media and are being secreted by the cells. Both of these can increase and decrease with toxicity, indicating that there are multiple mechanisms at play. The most common effect for thymidine is an increase with increasing concentration for the cardiotoxic compound. In contrast to thymidine, 2-deoxycytidine is decreased in the presence of cardiotoxic compounds. Both of these metabolites are principal nucleosides of DNA and components of the pyrimidine metabolism pathway. Changes in these metabolites reflect inhibition of mitochondrial enzymes necessary for mitochondrial DNA synthesis and mitochondrial replication. One of the ways that the quickPredict assay can be used during drug development is to understand whether or not a drug candidate is likely to cause structural or functional cardiotoxic liability based on the metabolite response. What we found when we were developing the assay was that lactic acid was largely an indicator of functional cardiotoxicants, and almost all of the functional cardiotoxicants caused an increase in lactic acid. In contrast, the other three metabolites were predictors of the structural cardiotoxicants. And so, based on which metabolite is changing at the lowest concentrations, you can understand, it can help you understand if a drug is more likely to cause a functional effect or a structural effect in vivo. And this data could be used to understand differences in compound classes, such as with tyrosine kinase inhibitors. A number of tyrosine kinase inhibitors have been tested in the assay, and these compounds are known to cause a wide range of cardiotoxic effects in vivo, ranging from QT prolongation to congestive heart failure. The data from the assay shows that tyrosine kinase inhibitors are inducing a wide range of metabolic changes. 7 of the TKIs are changing metabolism for all 4 metabolites. However, the concentrations where an effect is seen for each of the metabolites can vary between TKIs. For example, crizotinib is increasing lactic acid metabolism at concentrations lower than where changes are observed for the other metabolites. Imatinib, lapatinib, sorafenib, and vandetanib are all changing arachidonic acid at the lowest concentrations. Nilotinib and sunitinib are causing a change in thymidine metabolism at concentrations lower than the other metabolites, whereas dasatinib and erlotinib are causing changes in 2-deoxycytidine at the lowest concentrations. So what does that all mean? Well, as I had mentioned, compounds that impact viability in lactic acid at the lowest concentrations are likely to cause functional cardiotoxicity or proarrhythmia. Interestingly, one of the effects of crizotinib, which impacts lactic acid at the lowest concentration, is QT prolongation. Drug-induced dysregulation of thymidine and 2-deoxycytidine indicates that the compound's interfering with mitochondrial DNA synthesis and replication, as I mentioned. 3 of the TKIs that we tested are inhibiting BRCA-ABL, which targets mitochondrial in response to oxidative stress and thereby mediates mitochondrial dysfunction and cell death. Interestingly, nilotinib and imatinib are increasing the levels of thymidine, whereas dasatinib is inhibiting 2-deoxycytidine, indicating that while these all target BRCA-ABL, they are likely acting through different pathways. And the compounds where you're seeing an effect in arachidonic acid first are likely indicating that the compound could be causing cardiotoxicity through dysregulation of lipid transporter metabolism, fatty acid metabolism, or a redox state imbalance. So in summary, the combination of the 4 metabolites used for quickPredict is able to predict the cardiotoxicity potential of 81 compounds with greater than or equal to 85% accuracy at 8 therapeutically relevant concentrations. Each metabolite can identify multiple mechanisms of cardiotoxicity, and the prediction model can be used to determine a concentration where a compound is likely to cause cardiotoxicity. And one of the things I didn't touch on much today, but I'd like to point out, is that this method can be combined with other assays and endpoints for a comprehensive understanding of a compound's cardiotoxic liability. And currently we are working with the hESC group on their multi-site study to identify chronic treatment-induced cardiotoxicity using in vitro assays. I'd like to thank my group at Stemina and my coworkers there for all of their hard work on the research that I've presented here today, as well as my collaborators at the EPA, Tom Knudsen and Todd Zerlinden. At Imperial Tobacco, Liam Sims and his team, in particular, I've worked with Catherine Rudd and Fan Yu, and then Christine Glatt, who I worked with from the Chemours company on the Gaseous Chemicals study that I discussed. I'd like to thank everyone for attending or listening to our sponsored session today, and remind you to stop by our booth if you are interested in learning more about any of the studies discussed here. I know that I didn't go into great detail in the interest of time, but I'd be happy to discuss any of them with you in more detail. And also to check out the posters on our devTOX assay, poster 108 and poster 106. Thank you, and I hope everyone has a great day, and I'll be sticking around for questions.